Good afternoon, everyone. Thank you so much for joining us for the Cell and Gene Therapies Innovation Showcase. I'm Shana Kelly. I'm a faculty member at the University of Toronto and also the director of Prime. I'll be moderating our session today. This is a two-day event. First day was last week. Now we're moving on to kind of the enabling technology side of cell and gene therapies. We have a really exciting lineup for you today. But first, let's just talk about a few housekeeping notes. We are going to record today's session so that folks that can't join us right now can view the presentations later. Live captioning is enabled. You can see the captions if you click the live transcript button at the bottom right of your screen. The chat, because we have a fairly large group joining us today, it's been disabled. Use the Q&A box to leave your questions. We're going to have all the presentations kind of back to back, and then we'll have panel discussions just to go through the questions that you bring forward. We would love to hear from you. So please do type out your questions in the Q&A box. And so then before we get going, we should also just do our land acknowledgement. Despite the fact that we're doing this virtually, we wish to acknowledge the land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and most recently the Mississaugas of the Credit River. Today, Toronto is still home to many Indigenous people from across Turtle Island, and we are very grateful to have the opportunity to work on this land. So before we get going today, I just wanted to say a few words about Prime. So this event is being co-sponsored and was co-organized by Medicine by Design and Prime. You heard about Medicine by Design in the last session. It's a wonderful program that we have based here in Toronto, focused on regenerative medicine, very core application of cell and gene therapy. But today I'll just say a few words about Prime before we get going. Prime is an initiative based at the University of Toronto. It is focused on next generation precision medicine, the development of new therapeutics, diagnostics, new ways of looking at disease biology across many different indications. It brings together over 80 faculty members from 4 different faculties at U of T, from 15 different departments. We also have 150 trainees. And our mission at Prime is to really elevate research, increase the research funding that our campus sees in these areas, really empowering collaborations, training a new generation of multidisciplinary and translational leaders, creating community. We have a large ecosystem in Toronto. We really want our community to be as connected as possible. And we also are quite focused on building Toronto's biotech ecosystem. So that's Prime in a nutshell. This is a very translational and entrepreneurial group of people, lots of companies started, venture capital raised, all with an eye towards impact in the biomedical sciences. I won't spend too much more time because we're kind of ready now, I think, to move on to our next speaker, but hopefully that gives you a sense of what we do at Prime at the University of Toronto. So then without further ado, I think I will introduce our first speaker. Christine, yeah, if you want to go to the agenda. So we're running this from 1 to 3 today. As I mentioned, we're going to have back-to-back presentations. We'll have some discussion, and then we'll move on to a second set of presenters and then have some discussion with them, and then wrap up right on time at 3 o'clock. So for our presenters, I'm going to turn my camera off, but if you're going over time, I'm going to turn it back on so that you are aware that your time is up, and we just need to keep things on time to the extent possible. So without any further ado, I will introduce our first speaker today, who is Maud Dumont-Lagasse, and she is the Associate Director of R&D and Special Products at XcelThera, which is a Montreal-based company developing bioengineered solutions for blood cancers. Maud completed a PhD in molecular biology with a specialization in immunobiology. at UWM before joining Excelthera in 2018. Today, she's going to present work done in close collaboration with the hematology team leading clinical trials at Hospital Montreal. And this work explores how cord blood expansion increases donor availability for different populations. So please take it away, Mon.
Thank you so much for this introduction. So I would first like to thank the organizers for giving me the opportunity to talk about the work we do at Excelthera. It is a highly collaborative work, as mentioned by Shanna. So I'm just going to share my screen right away, and then screen 2, and then present. Let me know if you see my presentation. I think it should be fine.
We don't. It says that you're screen sharing, but that you need to double click to enter full screen mode.
Meeting controls.
All right, just bear with us for a moment, everybody.
Okay. Can you see it now?
Now, yes, if you go into full screen mode, we should be good to go.
Is that okay?
Perfect. Go for it.
Okay, thank you. All right. Okay, so Excelthera is a cell therapy company based in Montreal that was created after the discovery of the UM171 molecule by the laboratory of Dr. Sauvageau and Dr. Marinier at Université de Montréal. So the molecule was developed and optimized between 2010 and 2013 for its property to expand stem cells, hematopoietic stem cells ex vivo. Following the good results that they had, they decided to create Excelthera in 2014 and continue with preclinical development. And the first clinical trial using the UM171 cord blood transplant was initiated in 2016, including mostly patients with hematological malignancies who lacked a donor. So if you're interested in the full trial results, they were recently published by Dr. Cohen in the Lancet Hematology in 2020. So before we go to the results of the trial, we might ask the question, what is the use of expanding cord blood stem cells? So hematopoietic stem cell transplantation can be performed from 3 different stem cell sources. The most commonly used are bone marrow and mobilized peripheral blood. So these 2 sources of stem cells are collected from adult donors that are specifically matched with the patients. They represent the standard of care for HSCT. However, due to the maturity of the cells contained within either bone marrow or peripheral blood, the donor requires to be very stringently HLA matched to the patient. This makes matching more difficult between donor and recipient and leads to several months of delays between diagnostic and transplant for a lot of patients, but also to a significant proportion of patients having access to no donor, basically. So that's where cord blood becomes interesting. Cord blood can be collected at birth, so from the umbilical cord blood, without any risk to the babies and the mothers. And due to the immaturity of the cells that they contain, they require less stringent HLA matching, which makes it easier to find a match between donor and recipient. So historically, the minimal HLA matching that was recommended was 4 out of 6 HLA genes or HLA alleles. And with more precise allele sequencing, now we translate that into 5 out of 8 HLA matched requirements. So cord blood units are cryopreserved in cord blood banks worldwide and are readily available for transplantation, which is a huge advantage as patients waiting for a transplant can see their disease worsen if they wait too long, if they have to wait too long, basically. The bad side of cord blood is that most cord blood units contain a low number of cells, translating into a low stem cell dose for adult patients especially. And this has been associated with several transplant-related complications. These complications can be summarized as delayed engraftment and immune reconstitution, leading to an increased risk of graft failure and increased infection incidence and severity. These both combine to increase the risk of transplant-related mortality, making this graft a little bit more unsafe than what we would see with bone marrow or peripheral blood. And so that's why expanding cord blood stem cells appears as an attractive strategy to prevent the low cell dose issue of cord blood. So that's where Exceltera's technology comes in. The manufacturing process is quite simple. We basically select a single cord blood unit that is specifically matched to a patient that was recruited within a trial. And once we receive the unit, we can thaw it and perform CD34 positive cell selection. So the separation of CD34 positive cells from the rest of the cells contained within the graft is done through immunomagnetic bead enrichment using the Clinimax system that is approved for clinical use. The CD34 positive cells are enriched in hematopoietic stem and progenitor cells. They will be placed in culture in the presence of UM171 for a period of 7 days with other growth factors. And after this 7-day culture period, the cells will be washed to remove the culture reagents and the UM171 molecule. And only the cells will be formulated and cryopreserved for future use. The CD34 negative fraction, which contains more mature cells, so including the T cells, are also prepared, formulated, and cryopreserved, but there is no culture involved with these cells. And both products are infused together to the patients and are important to provide prompt hematopoietic reconstitution and early post-transplant immune protection. Within our trials, we have seen very encouraging results with a prompt engraftment. We saw the 100 neutrophil per microliter threshold attained quite early, so at 9.5 days, which is earlier than what is seen with other stem cell sources. And patients also benefited from very quick resolution of fever. So compared to cord blood or bone marrow expansion, there was a significantly lower time to fever resolution. The grafts are well tolerated, leading to a fast immunosuppression withdrawal, with most of the patients being off immunosuppression withdrawal at 1 year, which is not the case with standard of care, basically. All of these combined to very good, what we call GRFS. So GRFS is GVHD and relapse-free survival. It is a metric that encompasses all the important outcomes following a transplant. So patients who succeed with this metric actually do not have a grade 3 to 4 acute GVHD, moderate to severe chronic GVHD. They don't relapse and they survive basically the transplant. And compared to a cohort of 907 patients transplanted at University of Minnesota, which is shown in gray here, the patients transplanted with UM171 expanded cord blood showed very good GRFS. The UM171 grafts show a very different cell composition. So in a study where that I led, where we analyzed the immune reconstitution in patients, in our patients transplanted with UM171 grafts, we compared our patients to a cohort transplanted with unmanipulated cord blood. So which you see in red here, in our patients in blue. And while the number of total nucleated cells was quite similar between the two groups, there was no significant difference. The cell composition differed greatly. So we had a 17-fold higher dose of CD34 positive cells, which contains the stem cells, thanks to the UM171 expansion. But we also had a lower dose of CD3 positive cells. And this is due to the manufacturing steps necessary for the separation of CD34 positive cells for the culture. So we wondered if that might translate into different clinical results, basically. Interestingly, the T cell reconstitution was quite similar between the two cohorts. So here I show CD8 and CD4 T cell counts at 3, 6, and 12 months in both cohorts. And basically what our results show is that we didn't see any differences in the numbers and the phenotypes of the T cells. So the lower T cell dose infused with the UM171 grafts does not seem to result in any delayed T cell reconstitution. We decided to go a little bit further into the analysis of T cells by performing deep TCR sequencing. And the results were quite interesting. Basically, the UM171 patients present a higher T cell diversity at 12 months. So this can be shown by the number of unique T cell clones that can be detected within a sample, but also with metrics of diversity, such as the Gini index, which is presented in the paper that we published in Stem Cells Translational Medicine. Interestingly, the increase in T cell diversity was most pronounced in younger patients, so patients under 40 years old, which was also accompanied by an increase in naive T cells and recent thymic immigrants, suggesting that the UM171 expansion might actually lead to an enhanced thymopoiesis in younger patients. This is correlated with an increased common lymphoid progenitor frequency and potency within our grafts. So these are in vitro assays, but basically what we've observed is that the concentration of common lymphoid progenitors is increased with UM171 cultures, but also for each CLPs, or common lymphoid progenitor cell, plated, the number of cells contained within a colony forming unit was higher, meaning that they were more active. So that might be one explanation to the enhanced thymopoiesis that we observed. And how this translated for the patient is actually better immunity. So we compiled the number of infections and the severity of each infectious events that occurred within our two cohorts. And basically what we saw is that the UM171 patients, shown in blue here, benefited from an early protection against bacterial infections, both grade 2 to 3 and grade 1 to 3. And this was particularly clear in the early post-transplant period, the first month after transplantation, where the patient is most at risk, actually. We also saw that the viral infections were lower for the grade 2 to 3 viral infections were lower for patients transplanted with UM171 cord blood. So this means that not only in terms of T cell numbers, we have a similar reconstitution, but in terms of T cell diversity and T cell function, we seem to have an advantage linked to the UM171 grafts. Finally, one of the very important aspects of the UM171 expansion is that it increases the pool of suitable cord blood units that are available for patients. So here I show 3 graphs. Basically, it's the same data set, but adapted for different patient weights. So the minimal CD34 and TNC cell dose required for single unmanipulated cord blood or double unmanipulated cord blood transplantation makes it harder to find a donor for patients, as only a very small proportion of the cord blood banks are actually available for these patients. By allowing the use of smaller cord bloods, UM171 expanded grafts actually increases the proportion of cord blood units that contain sufficient cells for patients, meaning that for instance, for a 100-kilo patient, if a physician would like to perform a single unimpeded cord blood transplant, he would have to choose within 0.5% of cord blood units stored within a cord blood bank. While with the UM171, the physician would have access to 23% of cord blood units, which makes a big difference, basically. And this higher donor availability also translated into better HLA matching within our trials. And so for our first phase 1/2 trial, we've seen that approximately 50% of patients had access to a better HLA match cord blood than what would have been possible without expansion. And more than 75% of them had 6 or 7 out of 8 HLA match units, which is very good.
Todd, we'll need you to okay, you were.
Yeah, I'm done.
Thank you.
And so basically, in conclusion, our results suggest that the UM171 expanded cord blood grafts are safe and effective, leading to low transplant-related complications and providing good immune tolerance. The immune reconstitution is prompt and robust. We had high T-cell diversity at 6 and 12 months post-transplant, with a better immune protection against bacterial and viral infections. And basically, by allowing more access to more cord blood donors, the patients also benefited from better HLA matched units. So I briefly want to thank all the people that have been involved within these studies, and I will be happy to take questions at the Q&A sessions at the end of this group presentation. Thank you.
Okay, thank you so much, Maud. We will come back to you in a bit with some questions. For now, we're going to move on to our next speaker, Rony Chidiac, who's a postdoctoral fellow in the lab of Stéphane Auger in the Leslie Den Faculty of Pharmacy at the University of Toronto. Rony has a PhD in pathology and cell biology from University of Montreal. His current research is focused on modulating the Wnt pathway using novel antibody-based agonists that selectively target frizzled receptors as a path towards regenerative medicine therapy. Rony was one of our 2019 Prime Fellows and recently got a really nice new fellowship from the Bright Focus Foundation. All right, Rony, take
Thanks,
it away.
Luciana. Can you see my slides? Can you hear me?
Yeah, looks good.
Okay,
Go ahead.
perfect. Thank you so much. And I would like also to thank the organizer for the invitation. So it's a great pleasure for me actually today to talk to you about our recent work in the lab, introducing the Frizzle-4 LRP5 antibody as a drug candidate for the treatment of the vascular retinopathy. So when beta-catenin signaling actually is required for the formation and the maintenance of the blood-retinal barrier, which is characterized by the formation of a tight junction between endothelial cells. Among the 10 different frizzle receptors, Frizzle-4 is expressed in the endothelial cells and act as a receptor for the secreted protein norrin. Norrin binds to Frizzle-4 along with LRP5 as a co-receptor and needs also the expression of the T-span12 as a third receptor in order to activate the Wnt beta-catenin signaling, which leads to the expression of some of the junctional proteins such as Claudin-5. A decrease in the downregulation of the Wnt signaling can lead to a decrease in the expression of the junctional protein, which leads to an increase in the vascular leakage. And this is a hallmark of multiple retinopathy diseases, such as diabetic retinopathy and retinopathy of prematurity, which could lead eventually to blindness. So genetic loss of function studies have shown that downregulation, knocking out NDP, LRP5, or Frizzle-4, or T-span12, or even beta-catenin can lead to a decrease in the barrier formation and the barrier function in the retina. However, interestingly, when these studies express a stabilized version of the beta-catenin in endothelial cells, this led to reverse the phenotypes that was seen in the Frizzle-4 and the norrin knockout, where the barrier function is promoted, and then there was a reverse in the phenotype. So there was enhancing and the barrier integrity in the retina. So that's led us to think that the pharmacological activation of the Frizzle-4 beta-catenin signaling in endothelial cells could represent a novel strategy to promote or restore the barrier function. And we think we have the right tool for that. So recently, we have a long collaboration with Dr. Sid, who is at the UFT, an expert in protein engineering, where we recently published in eLife in 2019, a paper where we introduced a tetravalent antibody as a novel tool to selectively and at will activate one or multiple Frizzle receptors. So these receptors act as a Wnt, similar to the Wnt purified proteins, so they activate very selectively the Wnt beta-catenin signaling. And knowing that Wnt signaling is downregulated in multiple diseases, we thought that this could be a valuable tool in order to selectively, at the receptor level, activate the Wnt signaling and could be very valuable for the regenerative medicine. And today I will be showing one of the first application of this tool using the Frizzle-4 and LRP5 antibody, since we know that Frizzle-4 and LRP5, as I introduced, are important for the barrier integrity. So Frizzle-4 LRP5, what we call is F4L5.13, which I will be referring in my presentation as the Frizzle-4 agonist, is also a tetravalent antibody. So it has a 2-diabody on each side of the Fc. On this side, you can see that it's very selective to bind to only Frizzle-4 among all the different Frizzle receptors. And the other side, it binds to LRP5, but not LRP6. So we have a molecule that can bind selectively to these two receptors, Frizzle-4 and LRP5. So we asked, the first question we asked is whether the Frizzle-4 agonist can activate the Wnt beta-catenin signaling in endothelial cells. So for that, we treated endothelial cells with either Norrin or a Frizzle-4 agonist, and we monitored the axin 2 mRNA expression by qPCR. And as you can see here, both Norrin and Frizzle-4 agonists can activate the Wnt signaling with Wnt beta-catenin signaling. However, our Frizzle-4 agonist has higher potency than the Norrin. So the next question, we know that Norrin depends on the T-span12 in order to activate the Wnt signaling. So we asked whether the Frizzle-4 is also dependent on the T-span12. Well, as you can see, as expected, if we downregulate T-span12 using an siRNA, this abolished completely the signaling induced by the Norrin. However, this was not affected by the treatment of the Frizzle-4 agonist. So we concluded that the Frizzle-4 and LRP5 clustering that is triggered by the Frizzle-4 agonist is sufficient to activate the beta-catenin signaling in endothelial cells, which is mimicking Norrin ligand. So one of the first earliest feature of retinopathy is VEGF-mediated breakdown of the blood-retina barrier, and VEGF increased permeability in endothelial cells. And that's why the standard care of use the VEGF therapy, anti-VEGF as a therapy for multiple retinopathies. So what we ask is whether our Frizzle-4 agonist can decrease or block the VEGF increased permeability in endothelial cells. So for that, we treated our endothelial cells with VEGF, and as you can see, this led to a decrease in the localization and the expression of junctional proteins such as ZO1 here, Claudin 3 and Claudin 5, and consequently this led to a significant increase in the permeability across the monolayer of endothelial cells. However, interestingly, when VEGF was co-treated with Frizzle-4 agonist, this completely blocked the permeability and rescued the phenotype seen with VEGF, and this in part due to the restoration of the junctional protein expression such as ZO1, Claudin 3, and Claudin 5, as you can see. So we concluded that the Frizzle-4 LRP5 agonist treatment can promote the endothelial cell barrier function in vitro. So what about in vivo? So we used two different models in order to test our Frizzle-4 agonist in vivo. The first model, we used a T-span12 knockout mice. So these mice display a defect in the blood-retina barrier, and they have a malformation in the vascular plexus. And so what we've done is we injected the mice with either an isotope control or a Frizzle-4 agonist starting from a P5 to P20. And then we stained for the retina with either isolectin to stain for endothelial cells or IgG to probe for barrier defects. And as you can see in the T-span, as expected, in the T-span12 knockout mice, you can see that there was a massive extravasation of the IgG, as indicating that the vessels are leaky. And also this there was a huge malformation in the vascular plexus. The deep layer was completely gone. However, when we treat the mice with the Frizzle-4 agonist, this completely rescued the phenotype. As you can see, that with the mice that are injected with the Frizzle-4, there is no leakage of the IgG, and the vascular plexus is not affected. So we even checked for the barrier function by the expression, by investigating the expression of the Claudin-5 and as a tight junction, and the PLVAP as an endothelial cell fenestration. As you can see, and as expected, in the T-span12 with the isotope control, you can see that there was a downregulation of the Claudin-5 and an increase in the PLVAP expression, indicating that there is a barrier defect. However, this was restored and completely rescued when we inject the mice with the Frizzle-4 agonist. So Claudin-5 is back again on the at the junction level, indicating that the barrier that there is a promotion of the barrier function. So we also use another model. So we tested our Frizzle-4 agonist if it can reduce the pathological neovascularization and the oxygen-induced retinopathy model, which is a model of diabetic retinopathy and retinopathy of prematurity. So what we did is that we kept the mice up to P7 in a regular room air, and then at P7 we moved the mice to a 75% oxygen chamber, where this could lead to a vessel loss. And then at P12 we inject the mice with either a vehicle anti-VEGF or a Frizzle-4 agonist, and then we move them back to a regular room air, where this will induce a neovascularization step, and then we sacrifice the mice at P17. And as you can see, our Frizzle-4 agonist can significantly decrease the reduce the pathological neovascularization, similar to what we got with anti-VEGF, which indicate that our Frizzle-4 agonist could be a tool to treat the diabetic retinopathy, for example. So the flag is a really powerful tool and very valuable tool. As I mentioned, Wnt signaling is downregulated or modulated in different diseases. So we can now treat selectively different Frizzle receptor in order to modulate the activation of Wnt signaling and try to treat these diseases. But what I've showed today at the level of the Frizzle-4 and the retina, we can use this as a promising tool in order to treat the retinopathy diseases. But the similarity between the blood-retina barrier and the blood-brain barrier led us to think that the Frizzle-4 can also work to treat the leaky vessels in the brain, which can cause also other diseases such as central nervous system disorders such as stroke, glioblastoma, and others. So for that, I would like to thank all the people that collaborated to this work, especially all the members of the Angers lab, the member of the Sidhu's lab, expert in the protein engineering, Jung's lab, which are expert in the vascular retinopathy models, and Therapeutic, which provided valuable resources founded by Stéphane and Def at the UFT, and also the funding agency, and for you for listening. Thank you so much.
All right, thank you so much. Beautiful work. So we'll come back with some questions in a bit. Our next speaker is Penney Gilbert. Penney is an associate professor at U of T in the Institute of Biomedical Engineering, and she's a faculty member affiliated with Prime. She received her PhD from the University of Pennsylvania, did a postdoc at Stanford before joining U of T in 2013. She's the recipient of an Ontario Early Researcher Award and holds a Canada Research Chair in Endogenous Repair. And today she is going to talk about preclinical models of human skeletal muscle function. So Penney, we'll turn it over to you. It looks like maybe you are there you go. Perfect. Take it away.
Okay, perfect. So thank you so much, Shanna. As Shanna noted, the focus of research in my lab is on skeletal muscle tissue. And skeletal muscle is the most abundant tissue in our body, and it's comprised of bundles of multinucleated cylindrical muscle cells that contract in response to human intent to enable functions like walking and smiling and swallowing. But there are situations where skeletal muscle integrity is lost, resulting in diminished quality of life, and in some cases, even death. One of these examples is Duchenne muscular dystrophy, which is caused by mutations in the dystrophin gene. And dystrophin is really a giant protein, and it bridges the specialized cytoskeleton that's on the inside of muscle cells to the proteins that are embedded in the plasma membrane of those muscle cells that then engage with the extracellular matrix outside to support contraction. And so without this protein bridge, DMD patient muscles decline in health with time, and this results in cardiorespiratory failure in the 3rd decade of life. And at the moment, corticosteroid treatment extends life by delaying disease progression, but this really does not prevent death. And so both gene and cell therapies are being tested as strategies to improve or extend life or even serve as a cure. And at the moment, mice and dogs with mutations in the dystrophin gene are commonly used to test promising DMD therapies. But what I'd like to talk about today is the possibility of humanized preclinical muscle assays to kind of bridge the gap between these animal studies and human clinical trials. So 2D cultures of multinucleated human muscle cells offer a really easy strategy to test batches of cells for myogenic tendencies or the ability to target dystrophin by gene editing strategies. And while 2D cultures offer a lot of benefits, studies of function are limited in this context owing to the detachment of the cells from the culture dish upon contraction. And so this is really where 3D cultures start to take center stage. They're really well suited to maintaining human muscle cultures for several weeks, lending to enhanced contractile maturity and allowing for studies of cellular function like strength and calcium handling. And in the case of DMD, it's contractile movement, so walking and running, that potentiates muscle cell membrane damage and leads to those unending cycles of degeneration and regeneration that are hallmarks of DMD. So the ability to study these features of DMD in muscle cultures is really contingent on these, the ability to stimulate contractions. And that was what motivated our interest in going down the path of trying to model DMD in culture. Now, the basic recipe for creating 3D muscle tissues in a dish is pretty straightforward. You need a source of transit amplifying myogenic progenitors. You're going to need a protein-rich extracellular matrix to suspend the cells within and act as a hydrogel. And you need some sort of culture device with a couple of attachment points, usually, that support the establishment of uniaxial tension. So those myogenic progenitors can be immortalized or primary cell lines that are derived from a patient or human muscle biopsy. They can be pluripotent stem cell derived or MyoD converted fibroblasts or from other sources. And then here I'm showing you one example of a very, very simple device where those attachment points that I referred to are literally just 2 pieces of Velcro at either end of a channel that is then filled with the cell extracellular matrix slurry. And what we find is that over a period of a day, those cells that were evenly spread in the well will self-organize to create a nascent tissue in the center between the 2 attachment points. And within a week, the tissue is populated with aligned and multinucleated and striated muscle cells, as you can see here. And they're contractile at this time point. So we have invented, or my team alone and also with collaborators have invented a number of different types of platforms with which to manufacture and study 3D muscle tissue function in culture. And so this first one that I'm showing you here is a 96-well flexible rubber device where it's possible to cast arrays of tiny little muscle microtissues that are across 2 attachment points that are flexible rubber posts, and the deflection of those posts can be used to quantify changes in tissue strength over time. This second example is an 8-well device that's made of hard plastic in this case, and can in theory be scaled to a 96-well format. And in this case, the advantage of the system is that it's an inverted design. And so in this case, the tissues are cast in close proximity to a glass cover slip. And hopefully what you can appreciate here is that the tissues are spanning across these 2 long, rigid, but long posts. And because they're long, the tissues, when they contract, will deflect those posts, and it's possible to get measurements of strength in the context of this device. But at the same time, because the tissues are close to the glass cover slip, it's also possible to do high resolution microscopy, which is a really nice feature of this system. And then more recently, we've been working with Alison McGuigan's lab, and we've adopted a very, very simple paper scaffold that supports both 96 well and 384 well footprint arrays of 3D muscle cultures for analysis. And so all of these platforms offer an opportunity, I think, to provide a quality control assay for therapeutic muscle cell batches, or to evaluate gene therapies using a humanized assay of muscle function. And each of these systems have their advantages and also their limitations. So it's context dependent. So what types of metrics are possible in this system? So I'll just highlight some of our recent work. We produced muscle microtissues using cells from DMD patients, and we established protocols to be able to assess the contractile apparatus using immunohistochemistry and electron microscopy. And we studied tissue strength and calcium handling downstream of stimulated contractions. And perhaps most importantly, in the context of DMD biology, we adapted assays of muscle membrane integrity that are typically employed in humans or in animal models of DMD to instead be implemented in our 3D cultures. And these included the creatine kinase activity assay, the Evans blue dye assay, and we have also employed electrophysiology to explore membrane integrity in these 3D cultures. And what I'll just point out is that very importantly, significant tissue level distinctions between healthy and dystrophic tissues only arose in our hands after induced contractile activity, which makes sense because this is also what we see in patients. So let me show you two very short examples, and then we can discuss more in the discussion period. So with our colleagues at UTM, my team prepared healthy and dystrophic 3D tissues, and then our colleagues conducted electrophysiological analyses of the single muscle cells that were in each of the tissues. And so here's an example of the data that's obtained using sharp electrode intracellular recording. And so in this case, we're looking at the resting membrane potential. Each of these dots is an individual muscle cell inside a 3D tissue. And the resting membrane potential is something that's determined by primarily two factors: the differences in ion concentration of the intracellular and extracellular fluids, and the relative permeability of the plasma membrane to different ion species. Now, in our body, a healthy muscle cell has a resting membrane potential of about -70 to -90. And so it's notable here when we look at these muscle cells that are made from healthy muscle donors, that we can find a resting membrane potential that is matching what we would expect in a human. And this is further notable because studies in 2D cultures that have tried to measure the resting membrane potential of human myotubes have not been able to achieve these sorts of resting membrane potentials that get so close to what we see in the human body. And then these two cell lines here are tissues that are made with DMD cells, two different patient lines. And hopefully what you can appreciate is the DMD myotube sarcolemma is significantly more depolarized. And that's suggesting that the membrane is leaky. And this is consistent with what we would expect from a DMD muscle tissue where you're missing dystrophin protein and that link to the ECM is disrupted. So as you'd hope, we can test the influence of therapies to modify the integrity of the dystrophic muscle cell membrane. So here is one of a few examples that we have where we treat the muscle cells, in this case, with a beta-1 integrin activating antibody. And that serves to enhance the ability of the cells to grab onto the extracellular matrix around them in the absence of dystrophin that would normally help to potentiate the forces through the cell membrane. And what we can see following this treatment when we look at the resting membrane potential is, here's the parental cell line and the muscle cells that are being tested. And you can see that upon treatment with either of these treatments, IgG4 or the beta-1 integrin activating antibody, that we see that the resting membrane potential becomes more negative. It's moving closer to this healthy control. And that tells us that the integrity of the membrane has improved downstream of these two treatments. I'll just note that the IgG4 treatment is one that is being tested in clinical trials in people. So next is an example of a phenotypic analysis where the outcome is contingent on stimulated contraction. So here we're looking at electron microscopy images of 3D muscle tissues from healthy cells here on the left and from DMD cells here on the right. These are two different patient lines. And hopefully what you can appreciate is that in the healthy cells, you have a nicely organized contractile apparatus with these dark black Z discs, I bands and A band structures. Whereas in the DMD cells, what we see are mostly aligned myofibrils. So you can see some of that and some degree of organization. So you see some remnants of banding patterns, but not to the level that's seen in the healthy tissues. And so then if we stimulate those DMD tissues to undergo a series of twitch and tetanus contractions and then conduct our electron microscopy analysis, we find that in the case of a stimulated contraction, that contractile apparatus shows up as just being scattered in all directions and is beyond repair. So this then creates a perfect starting place for a contractile assay, where then we're in a position to test treatments for the ability to both improve contractile apparatus maturation and to withstand the stress of induced contractions. And so here again, we're treating those DMD microtissues with either an IgG4 treatment or a beta-1 integrin activating antibody. And then we go on to induce a series of twitch and tetanus contractions on day 8 of culture, followed by EM analysis. And hopefully what you can appreciate is in this IgG4 treatment, we see that once again, the sarcomeres are kind of spread all over the place. But downstream of this beta-1 integrin activating treatment in these cells that lack dystrophin, not only do we see that the contractile apparatus has withstood the contraction, but the organization of the sarcomere is starting to look very, very similar to the healthy controls. We see these dark Z discs, we have the I bands and the A bands. So with that, hopefully I have convinced you of the merits of enlisting 3D muscle cultures as a humanized preclinical setting to test the effects of promising therapeutics and assays of tissue function. And as I mentioned, I really truly believe that these assays could offer a powerful strategy in the quality control of myogenic cell products and also in evaluating the functional effects of promising gene therapies. And rather than going into that in detail, I'll end here so we have a little time for discussion. And perhaps we can talk about that during the discussion period.
All right, thank you so much, Penney, and thank you to all of our presenters for keeping us more or less on time. It's challenging with these short slots. So we can now go through a couple questions. There's some in the Q&A box here, and I would encourage our attendees to send any others along that you would like answered. We'll start with one for Maud, which is what makes HLA matching less stringent when derived from cord blood?
So the current theory is really that the immune cells contained in, I don't think you can see me anymore, but I think you can hear me. Got some problems with my camera, sorry. The immune cells contained within cord blood units are more immature than what you find in an adult. And this leads to more tolerance against HLA mismatches because the T cells are less mature, less educated or selected. There also might be another contribution of the low T cell dose basically that are contained within the cord blood grafts. So when compared to what a patient would receive with bone marrow or peripheral blood transplantation, I think the amount of T cells that they will receive is 100-fold lower, which might also decrease the risk of graft versus host disease. So yeah, basically that's the reason why cord blood is more tolerant to HLA mismatch in a gist.
Okay, great. Thank you so much. this is a question for Rony. How long does the flag treatment last? And from a therapeutic perspective, would it be a one-time dose or continued administration?
Yeah, so it's a very good question, actually. The flag are antibody-based agonists, so the half-life of the flag is similar or even better in vivo compared to IgG, which is around 20-30 days. But in terms of the therapeutic perspective in patients, like we know that for the T-span12 knockout, we were injecting every 2 days the mice, but was IP injection. For the OIR model, we were doing intravitreal injection one time. So we don't know exactly, like in a therapeutic perspective, if it's one injection or multiple injection. We hope that's going to be one injection, because comparing to the anti-VEGF, where it's very aggressive, you have to go like 3-4 times per year in order to get the anti-VEGF. We don't know exactly, I think, but hopefully it's going to be like better, and we hope that it's only one application, but we'll see.
Okay, fair enough. Thank you. Okay, and a question for Penney. This is just a question out of curiosity from me. You know, with these types of models, like the beautiful one that you've been developing, I guess there's always a question about how is the architecture affecting the cells? You know, is there anything specific that's coming from the interaction of kind of the template that you're putting the cells in? Have you guys looked at that at all, at the level of gene expression or just any other, maybe even phenotypic properties?
Do you mean in terms of the culture platforms themselves, or the extracellular matrix compositions that are used as scaffolds?
Yeah, I was thinking of the way that you're doing culture, kind of as opposed to just cells in a regular old dish.
Yeah, so I think at least from our standpoint, the biggest difference between just sticking muscle cells in a culture dish and putting them in 3D cultures, now you have ligation of the receptors on both the apical and the basal side. And you have a system that allows the cells to contract and those contractions to be maintained. And we know that those contractions are then translating to signal transduction in the cells, which potentiates the maturation of the cells. And ultimately, that seems to be the key to ultimately making a muscle cell that has a higher degree of contractile apparatus maturation compared to what you're going to see in a 2D culture dish. So it's just the types of questions you would ask in the two settings are just different. Sure. If you can ask it in a 2D culture dish, you should. It's not until you get to the point where you need to look at later stages of function that it makes sense to move to a 3D setting.
Yeah, okay, makes sense. Thank you. Okay, and we have another question from Maud. Is XCell Thera looking at applications for UM171 beyond cord blood expansion, maybe to other engineered cell products?
So we're currently trying to transpose our technology to adult stem cells, basically. So we know that there are some patients who need an autologous transplant, and some of them will be what we call poor mobilizers. So the number of stem cells that you can collect from these patients is very low, making it very risky to go to full transplant procedures with them. And so expansion of these cells might also be an interesting clinical use of the UM171 molecules. Yeah, that's the next step for us.
Okay. Okay, and there's one other question for you about the mechanism of action for UM171. Are you allowed to talk about that at all?
Yeah, there was recently a paper published, I think it was in Cell Stem Cell by the Sauvageau lab, basically describing a novel column 3 E3 ligase complex, which was crucial for the action of UM171 in basically regulating the epigenetic signatures, which are crucial for the maintenance of stem cells in vitro. So the paper is by Jalila Shagrawi. I invite you to go read it, and if you have any questions, you can always reach out to us. It will be a pleasure for us to answer your questions.
Okay, wonderful. Well, those are the questions. So what we'll do now is take a very brief break, 3 minutes, and then we will be back with our next round of speakers. But thanks so much to each of you for spending the time with us today. These were just fantastic presentations, really exciting science. So thanks again. And to all of our attendees, just stick with us and we'll be back in 3 minutes with the next round.
Thank you so much.
Thanks.
Thank you.
All right, well, it's 2 o'clock, so I think we should start things back up here. We've got a great set of speakers for our next session, and we're going to start with Sam Wadsworth. Sam is the Chief Scientific Officer and co-founder of Aspect Biosystems. He earned a PhD in respiratory cell biology from the University of Nottingham. He spent 2 decades driving developments in human tissue engineering. In 2013, he co-founded UBC spin-out Aspect Biosystems with Conrad Wallace, Simon Bayer, and Tamer Mohammed. And Aspect Biosystems, as you'll hear more about today, is a biotech company creating bioprinted therapeutics as medicines of the future. Sam is going to tell us about Aspect's proprietary bioprinting technology, and we'll talk about how it has the potential to revolutionize the field of regenerative medicine by enabling a new generation of allogeneic cell therapies. So Sam, we'll turn it over to you. I think you may just have one more button to click so that we have your slides kind of in there you go. Perfect. Take it away.
Great. Can you hear me okay, Shana?
Yes.
Perfect. Well, thanks for the introduction, and thank you to Medicine by Design and the prime organizers of today's Cell and Gene Therapies meeting. Thanks for the opportunity to present. And I'm looking forward to updating you on the exciting progress that we've been making at Aspect in creating bioprinted cellular therapeutics. So just to kind of summarize what we're trying to do here at Aspect, we're really focused on marrying our unique microfluidic bioprinting technology and manufacturing platform with advanced cell biology and biomaterials to generate bioprinted therapeutics. And these are really, what we think, the next generation of implantable cell-based therapeutics that we believe will unlock this next generation of cell therapies. So a bit of background on Aspect Biosystems. We span out from the University of British Columbia in Vancouver in late 2013. And it was really through a collaboration between two world-class research groups in medicine and engineering. Since we left campus and started to really ramp up commercial operations in around 2016, we've hit several key milestones. We assembled a world-class research team with over 50 scientists based mainly here in Vancouver, scientists, engineers, and entrepreneurs. We've built a strong intellectual property portfolio. We have over 50 filings around the world. We've created strategic partnerships with some of the biggest names in the industry. And we're now leveraging our bioprinting platform to create our own proprietary pipeline of bioprinted therapeutics. And so what is our technology? Well, at the core of our technology is our microfluidic bioprinting platform. And so we're the first and only company in the world that has really married the technologies of microfluidics and 3D printing. And what this does is it allows us to really have an unprecedented level of control over the materials that we're able to print with. With our platform, we can integrate, process, and print multiple different biomaterials that are loaded with therapeutically relevant cells. And the way that we print really enables us to print with really quite fragile cells. So we are able to generate tissues that have very high cell viability. We can print with stem cells, primary cells, either individual cells or clusters. And we can print with very high viability. And this is because the levels of shear stress that the cells are exposed to is very low. We're also able to not just switch between materials, but also combine multiple materials and multiple different cell types in the same fiber as we generate our printed tissue. And so we have very fine control over the positioning of cells and materials within our tissues at the fiber level. And so what you see on this slide is really an illustration of one of our print heads. I'm probably very small on the screen, but you can see I'm holding in my hand one of our microfluidic print heads. They're about the size of a USB stick. They have multiple channels in the print head. And what you see in this video here, if I can get it started, is just an illustration and a video of how the different materials are routed through the print head and combined into a single extrusion channel. And we're able to switch between multiple different biomaterials and extrude a cell-loaded fiber. But this is a 3D printing technology, and so we're able to pattern those cell-loaded fibers into 3D macrostructures. And it's these macrostructures that are our implantable cellular therapeutic tissues. So really what we see is that the ability to take a cell-loaded fiber that has multiple layers within the fiber can enable us to really develop allogeneic cell therapies. And this is because, obviously, if you have an allogeneic cell, it will be exposed to immune attack by the host after it's implanted into the patient. And so what we use here is we're leveraging our ability to generate multi-layered fibers. These fibers are only about the width of a human hair. The cells are embedded within the core of the fiber, and we have an outer shell that is a cell-free permeable membrane. And that permeable material enables the influx of growth factors and oxygen to the cells in the center of the fiber and keeps those cells alive. But it also provides protection from the adaptive immune system. And so the cells are protected from immune attack. And so this is really the fundamental technology that we're using to generate allogeneic cell-based therapeutics. Importantly, the patterns that we generate enable us to have consistent device function and also enable us to not just implant these devices, but also go back in to the patient and potentially retrieve the devices as well. Now, this kind of allogeneic cell bioprinted cell therapy platform is potentially applicable to multiple different diseases. We have our own therapeutic pipeline that we're working on in-house, and our initial area of focus is pancreatic tissue for the treatment of type 1 diabetes. This is basically where we're encapsulating pancreatic islets or beta cells into these core shell devices and using those as a treatment, developing these as a treatment for type 1 diabetes. We also have liver tissues in development at a slightly earlier stage, but again, these are designed around the concept of printing and encapsulating hepatocytes for the treatment of various different liver diseases, including acute liver failure and more chronic liver diseases as well. We also have several discovery programs in early stages of development as well. But today I'd like to talk about really the diabetes program that we have and focus in on some of the data that we're generating with this particular tissue. And so we've done a variety of in vitro experiments to demonstrate compatibility of our printing technology with pancreatic islets. But I'm going to show you some in vivo data sets that we've generated more recently. So what we do is, as I said before, we take these therapeutic cells. In this case, it's primary human pancreatic islets. They're embedded in the core of a core shell fiber. This fiber is about 1 millimeter in diameter. These fibers are, in this case, they're 8 centimeters long. We place them into the intraperitoneal cavity of mice. These are immune deficient mice in this case. And then after 31 days, we retrieve the fibers, stain them for live cells, and we can see that the green staining shows that there is very high cell viability. Each of those green circles is a pancreatic islet that shows high viability. The red stain shows insulin positivity via dithesone stain, and so these islets are also packed full of insulin and presumably quite functional, even after 31 days in vivo. The next experiment here was to test these tissues in diabetic models. And so we took these immune deficient NSG mice and treated them with streptozotocin. And you can see the trace on the bottom left of this slide shows blood glucose over time. We see a spike in blood glucose as the animals become hyperglycemic after STZ treatment. At 0 days, we implant with these 8 centimeter long fibers into the IP cavity that contain the primary human islets. And we get pretty rapid normalization of blood glucose. And that's maintained out for around 3 months in vivo when the experiment was terminated. This was with multiple different donors of pancreatic islets, and so we're confident that it's a consistent effect. This normalization of blood glucose is accompanied by a rise in human C-peptide as a surrogate for human insulin in these animals. And then on the right-hand side, you can see the kinetics of response. So this is where we give the animals an oral bolus of glucose. The black line shows the control animals that are non-diabetic, and you see a rapid spike in blood glucose that then normalizes fairly quickly. The dotted blue line is the animals that were diabetic that received the human tissues. And again, we see a very similar kinetics of response with the implanted human tissues, suggesting that the kinetics of the insulin release and subsequent control of glucose is physiologically appropriate in these animals. Now, a single fiber may be enough to control blood glucose in a mouse, but if we scale up to a human size, a fiber may need to be tens or hundreds of meters in length in order to load enough cells to control glucose. And so what we are doing is taking those fibers and patterning them into the 3D structures that I described earlier. And so on this slide, what you see is this is some in vitro data demonstrating how we can take a fiber that's a much longer length. We can pattern that into a 3D structure, and the cells within that structure, these are again primary human islets. The cells are viable for several days post-printing in vitro, and then we perform functional assays glucose-stimulated insulin secretion assays you see on the bottom right of the slide. This is where we take those printed tissues and we treat them with different concentrations of glucose. We go low, high, low, and we see a corresponding increase and then return to baseline in secreted C-peptide levels from these devices. The amount of C-peptide that we see that's released is proportional to the number of cells that we incorporate in the device, and it's almost a 1-to-1 ratio. So if we double the number of cells that are in the devices, we double the amount of C-peptide that's released, and that's shown in the graph on the bottom right. So this is how we're planning on scaling up the size of the device to human patients, but we also want to scale out. And so it's difficult to scale out using primary human islets. The number of donors is limited. And so we're also collaborating with a group at UBC, this is with Tim Kiefer's lab, to basically access stem cell derived beta cells. And this is some data showing in vitro functionality, again, using a GCIS assay of bioprinted stem cell derived beta cells. And then on the right-hand side, you can see a increase in circulating C-peptide when we implant these stem cell containing tissues into animals. And so we get increase in functionality over time in vivo. These experiments are ongoing. And so to kind of bring this back together, our goals as a company obviously is to bring together our biomanufacturing, our bioprinting technology with cells and biomaterials to generate these implanted 3D devices that can act as therapeutic tissues. We're demonstrating some really good functionality of these in vivo models, in small animals, and we're moving into larger animal models very soon. We also have a broad range of collaborators around the world through what we call our discovery ecosystem. And this is where we place our bioprinters into the labs of experts, and we work in various different tissue types, not just the pancreas and the liver tissues that I described. And so I'd be very happy to talk to people who are interested in working with us and collaborating. If you have a goal of developing cell-based therapeutics and you think bioprinting could help, then we'd be more than happy to talk to you. Please check out our website for more details. And then just at the end, I'd like to recognize the team that's behind all of this. We have a large, varied team here at Aspect. It's a very exciting place to work, and we have multiple different open positions that we're hiring for at the moment. So again, if you're interested in learning more about potentially helping us to develop these life-changing therapeutics, please just reach out on our website or to me, and I'd be happy to talk to you about this. With that, thanks very much, and I'm happy to answer questions in the Q&A period later.
Thank you so much, Sam. That was fantastic, really exciting. So we'll come back to you for some questions at the end. Next, we are going to move on to Maria Abduqatra. She's a trained biologist with a PhD in computational and mathematical biology from McMaster. She was a postdoc at the Max Planck Institute for Evolutionary Biology in Germany, and she is currently a research associate in the lab of Gary Bader, who's in the Donnelly Center at the University of Toronto. She's been with us since 2016, and she's going to tell us more about her work developing 3D mathematical models that explore cell development. Go ahead, Maria.
Thank you. I'm just going to start by sharing my screen. You can see my screen now, right?
Yeah, but I think you just need to click maybe to swap the displays.
So, which display? You see my, like, where my mouse is moving?
We see the, like, the presenter mode. We don't see the full screen.
I'm sorry, I'll do that again. Okay. So,
there you go. Perfect.
Sorry about that. And hi, everyone. Thank you, Medicine by Design and Prime, for the invitation and, of course, giving me the opportunity to talk about our work today. So I thought I'd start with a vision, at least my vision, and the possibility for a future where you can think of a patient can start and activate a specific treatment with just a simple push of a button or a thumbprint. And this vision involves what others are calling the digital twin. So it's a virtual proxy that can be used to analyze and test precise drugs regime as an example. This is, of course, a vision, but we have the building blocks already in place for a future with such possibilities. We have the experimental tools, data, and artificial intelligence already exchanging information and setting up the platform for learning and making these predictions. However, in my opinion, we are still missing another important building block, and that is the mechanistic models that use mathematical and physical sciences. These models can help provide orthogonal information that may direct our decisions and perspectives. They can even act as proxies to experiments and help us push boundaries outside of the accessible zone. So I think by working together and tackle problems using these various tools, we can forge new discoveries and maybe make way for this digital twin. Obviously, I have no idea when we will get close to our digital twin, but what I can tell you today is that we are close to what you can call the digital organoid or digital cell culture right now. So you can add a virtual version of these tools, and this can change maybe the way experiments are developed and designed. Models can guide the experiment and can make initial predictions before you start. So to develop these virtual cells, we start by integrating knowledge from many disciplines. And as a platform, it is powerful, modulable, and fast. For example, here, I'm showing just a platform where we chose a time as developmental days. You can have death probability, density, and any other cues that were associated with cell behavior and cell decisions. This is what we call rule-based agent models. The rules can be explored individually or in combination. And the rules we apply in the model, they can help generate emergent behaviors. So these help make predictions. We can track lineages across 3D space and time, and all this is done at any resolution. So in this case, it's the single cell level. So we can then apply the same analysis that is done on the current single cell experiments, for instance. Basically, the creativity lies in figuring out how to translate our knowledge from text to algorithm, and then to decide what to include and exclude in the system. But what happens when we ask a question and there is no answer, or there is a gap in our knowledge? This is where models can also do more than just make predictions. They can help fill missing information. This was the case when we started building the model that aims to disentangle how a single cell generates the diversity needed to create a living organism. We started integrating the algorithm that captures cell division, because development pretty much starts with a lot of divisions. However, how fast do these cells divide? Is it a constant rate across development? What is a typical cell cycle duration? We just kept on asking questions and quickly learned that we are missing knowledge. And this led to us to test the hypothesis that cell cycle duration is doing more than just controlling cell numbers. We learned that the cell cycle defines the time a cell has to grow, interact with its environment, duplicates the genome, and so that 2 copies are ready before it divides into 2 new cells. This is canonically defined by 4 phases. You have the 2 gap phases, an S phase where the DNA replication occurs, and the M phase where division occurs. And it turns out cell cycles start relatively fast and then slow down. And this is consistent across all multicellular eukaryotes in embryogenesis. And then we learned early on, these cell cycle durations are not just fast, but they also lack the time for growth and transcription. And this is important because cells use these genes to communicate and define their identity. In some species, we learned that, such as Drosophila, short cell cycles can be as short as 8 minutes long. And this is not enough time to complete some of the longer genes needed, which are known to take long transcription time. We also learned that a lot of the genomes carry a broad distribution of gene length. For instance, in humans, genes can range between 55 base pairs to 2.5 million base pairs. So we integrated this information in the model. And for this example, we designed a platform that tested the intrinsic effects of cell cycle duration. So we didn't include anything external. We ran different experiments. In one of our experiments, we started with a parental cell, and we allowed that cell to divide. In this case, it generated 2 identical cells, which then generated the same number of cells and the same kinds of cells. When we changed the scenario and we allowed this parental cell to divide and now make one fast lineage and one slow lineage, we find that the slow cells generated a population with a greater cell diversity. However, when compared to the faster cells, this came at a cost to cell number. There was a lot fewer slow cells in the system. We could also run other experiments that test what quenches such effects. We found that the typical stem cell-like scenario, so where a parental cell divides and generates 2 different cells, but this time, only one of the cells is allowed to divide further and generate offspring. In this scenario, we can overcome the cell number constraint. So we see a lot more slow cells in the system, but again, it comes at a cost. This time it reduces the number of fast stem cells in the system. We ran many other simulations which helped us generate a new theoretical framework. And from this, we moved on and started to ask questions. What happens when we add other parameters or test different known information that we know affect cell decisions? In this case, we asked what happened when we add gene-gene interactions from this perspective. So I will show you one example, and we start with just a 2-gene system, and we assume that X is 5 times longer than Y. And here, just to explain, the time on the axis is cell divisions. Here, the frequencies show us the gene frequencies, the average gene frequencies across time. Each row represents simulations, and it shows you how the cell cycle changed in time. Now, when we include interactions, in this case, it's a repressive interaction, so X represses Y and Y represses X, we see that since Y is shorter, it's going to be transcribed faster, and it's going to take over much quicker in the system. So irrespective of what the cell cycle says, the gene length in this case moves the system and pushes Y. So we'll see all the cells expressing Y. However, in another example, when we see that where X represses Y, the cell cycle affects the system. In the first row, we have simulations where cell cycle duration is increasing, and we see a state where most cells will exhibit gene Y. And just once the cell cycle duration increases in time, you will see some of the cells now expressing X. If cell cycle duration is constant and long enough to express both genes, we'll see X take over. And then in a really interesting scenario, when you see the cell cycle duration decrease in time, we actually observe fluctuating. Early on, the cells will exhibit high Y, then you'll have, in the middle, we have most of the cells are exhibiting X, and then it switches back to Y because now the cell cycle is too fast to express Y, to express X. So we do not know exactly what all these mean, but what I hope to show you is that these simple parameters, like gene length and cell cycle durations, have been assumed to play a minor role in cell differentiation. While our model showed us that cell cycle duration not only affects cell number, but it can fine-tune cell number, cell proportion, and gene expression. And in combination with gene length and gene interaction, this may be a fundamental process in cell lineages and fate decisions. So as we build our platform, we hope to integrate our new knowledge. And this will hopefully motivate more experiments and help us learn how cells make decisions and teach us to better control them. I hope I showed you a glimpse of a future with models, but I also hoped to show that the present strength of models as tools can test hypotheses and help fill our missing gaps in our understanding. So I'd like to thank you guys for listening today. My supervisor, Gary Bader, Ruth, and Brendan, and the Bader Lab for their constant support, and especially Nika, Derek Vandergoy, and Peter Zanstra for their constant listening and willing to listen to our ideas. Thank you.
Okay, thank you so much, Maria. That was terrific. We'll come back to you with questions in a bit. and move on to our next speaker, who is Liliana Attisano. She's a professor and interim chair in the Department of Biochemistry at the University of Toronto. She has a lab based in our Donnelly Center. She's a faculty member affiliated with PRIME. Her research is focused on studying the molecular events that underlie signaling cascades, and she also works to elucidate how cells interpret contextual clues to control complex biological responses. Today, she's going to tell us about a new in vitro platform for the vascularization of human organoids. Take it away, Lil.
Thank you very much, Shauna. So it's my pleasure to be able to speak at this session, being a member of both MBD and PRIME. Having the opportunity to speak to both groups is terrific. And what I'm going to focus on, as Shauna said, is our efforts to develop an in vitro platform for vascularization. Okay, so I think most of us are aware, and has been showcased in the talks today, that there's been a shift towards using human 3D tissue models for research and therapeutics. And this has, of course, been enabled by our increased understanding in the biology and the ability to generate tissues, but is particularly exacerbated by the knowledge that human tissues are not the same as our favorite in vivo model, which is mouse. And this is nicely illustrated in the case of the brain. So these brains here, human and mouse, are drawn to scale. So not only is there a huge difference in size, but they also look different. So here's a zoom in view of the mouse. And you can see that structurally, it's quite different than the human brain, though of course, many of the basic developmental features are conserved. And so there has been, there was a huge effort to try and develop brain organoids, and including some work that, from my own lab, though we were following in the footsteps set by others before us. And here's an example of a human stem cell or derived cerebral organoid at 8 weeks old, developed in the lab. And so having, I'm going to just remove the picture. So having these models, we can now begin to address a variety of different questions. You can try and use them for organ replacement therapy, disease modeling, or drug efficacy testing. However, it was realized early on that one of the limitations of being able to do this is the huge heterogeneity that's present in different generated organoids. And this was really nicely illustrated in the case of brain organoids by Lancaster and Noblick, and they provided a little guide when you're making it. If it looks like this, just discard it, it's not going to make it, that kind of thing. And in fact, this heterogeneity and failure still exists in many different tissue models. So this is something that still needs to be addressed. But we were interested in modeling human disease. And so it was important for us to try and get rid of this heterogeneity. And Adam Civitilli, a grad student in my lab, took on this task. And he basically developed or modified the Lancaster protocol such that when he made the organoids, each one was very uniform. So here's his protocol. Here's what you often see using the traditional protocol. So not only were they the same size, they developed at the same time course, and single cell sequencing importantly demonstrated the repertoire of different cell types was constant from one organoid to another. So if you wanted to study a disease process, you need, and the emergence of different cells, you needed to be confident that they were reproducible. And so he was able to achieve this. And of course, we've begun to address a variety of studies using these COs in collaboration with many other labs. But we soon realized that there was a second critical problem. And what I'm showing you here is a cerebral organoid that's been put in slice. And in the center, as they develop, the core becomes necrotic. This is, speculated and modeled in this paper that basically shows cells in the center don't get enough oxygen or nutrients, so only the outer layer, or outer circle is viable and proliferative. But we need to keep the COs in culture for months, up to a year, in order to fully develop the neuronal types we'd like to see. And so this necrotic core caused a problem. First hints that this could be overcome came from Fred Gage's lab, where he took a cerebral organoid, implanted it in the brain of a mouse, and was able to vascularize it, and it continued to develop. And so the question we posed, can we introduce vasculature in human COs? And this is a project undertaken by Adam and Tiger in collaborative manner with my lab, Laurence Pelchet's, and Jeff Rana's lab. So bottom line is we did have success in this endeavor, and I just want to show you the key features. So at the center is a microfluidic chip, and in the center is a hydrogel in which we would implant the tissue of interest. There's a pump that will pump fluid through the chip and in a reservoir. The device is placed in a 6-well dish, 3 devices per dish, with the media reservoir in the alternating wells. This is a picture of what it looks like in the incubator. This is 1 unit of 3 devices. So the way we vascularize the tissue is simply mixing, not simply, but we mix endothelial cells, fibroblasts, and pericytes, which are required for vessel formation. Add in the tissue if you want, pour it into the hydrogel, and then turn on the pump. And this is sort of a schematic of the side view. This is the inlet. This is the hydrogel with the tissue. This is the outlet. And after a week, you can get the vessels formed. Here's some images of the fixed vessels. These are fixed and stained with CD31 to mark the vessels. And so you can see at 7 days, we have this nice vessel formation. And if we perfuse them with a fluorescent dye, you can see that the dye is contained within the vessels so that they're intact. I can also show you this in another image, this time using RFP labeled brain microvascular endothelial cells. On the left is the cells just at various stages of development. They're yellow, but they're still the RFP. And now I want to show you here at day 20. And what we've done in this panel is we've added in small fluorescent beads into the inlet and then turned on the flow. And what you can see is the beads flowing through the vessels. If you take a time-lapse images of the bead flow, this is actually a different experiment, but you can see that the liquid flow along the vessels is in a quite smooth manner, similar to what you might see in the blood. Okay, so I've talked about the vessel formation. Now, what about what happens when you implant a cerebral organoid? Okay, so this there's a lot of stains here. Pink is the endothelial cells. Green is the dendrites, so it's marking neurons. GFAP marks astrocytes, and SOX2 are the neural progenitor cells. So we have the inlet here, and then we move into the hydrogel, and you see the vessels entering into the cerebral organoid, penetrating and coming out the other side. Here's sort of a 3D rendering of a 2-photon image that we took. And remember, pink is the vessels, and you can see the vessels penetrating into the cerebral organoid, and we can see both progenitors and mature neurons in the vicinity. Okay, a key feature of the vessel formation in the brain is what's called a neurovascular unit, which is comprised of endothelial cells, pericytes, which coat the vessels, and then you have astrocytes, which make contact, and then you have your neurons in the region and microglia. So I just want to show you some evidence that we feel we are forming a neurovascular unit. So here's the vessel here, and you see green are the astrocytes and blue are the neurons that are surrounding the vessel. And I'm going to show you a slice, which is one of my favorite images. You see the vessels here, and the green are the astrocytes. And if you look here, you can see what looks like what's called an astrocyte foot, where the astrocytes make direct contact with the endothelial cells. So we evidence that the neurovascular unit is forming. Sorry, I went through a bit quickly. So the one thing that's missing in the cerebral organoids, even with the vessels, is sort of an immune component. And a student in Jeff Rana's lab, Amin, decided to ask the question whether he could incorporate microglia into the vascularized human cerebral organoids. So he took human embryonic stem cells, differentiated them into hematopoietic progenitors, because that's the origin of the microglia. They don't arise from the brain itself. And what he was doing before vivos was simply mixing the two, putting them in co-culture, and then letting them grow. And so he asked the question, if he mixed them and implanted them into vivos, what would happen? And so I'm going to show you some snippets of data from his work. So what he used GFP labeled stem cells. So the microglia, or proposed microglia, are green. And if he puts them in spinning culture, you get these little, this is a low mag image, a little tiny dot of what are microglial cells. If you put it in vivo, so in the vascularized chamber, there's a huge increase in the number of microglia. And this is quantitated here on this plot. But more importantly is that in the vivos, with the vasculature, the microglia became ramified. So an indication of maturation of the microglia. And here are 3 example shots in vivos versus those in spinning culture. Down in the bottom, he's confirmed that in vivos, they express IBA1, which is a marker of microglia. And so that's stained in this panel. Whereas in spinning culture, they are still microglia, they're IBA1 positive, but they just retain this round, immature phenotype. And more excitingly, and this is rather new data, is he also stained for CD163. And this is not specific for microglia, but importantly, perivascular microglia express CD163. So this suggests to us that we are getting a neurovascular unit formed, and that this allows for activation of CD163 expression in this population of microglia. So we still have a ways to go, but just that without the vasculature, this phenotype would never have been manifested in an in vitro system. Besides the brain, which is where we started, we have also implanted other tissues. These are some early experiments. So this is a human a stem cell derived intestinal organoid, and we've also implanted a human fetal retina, and we can see that the vessels form fine, independent of the tissue. Though of course, what cell types might emerge in different contexts is a subject of intense interest for us. And we're also moving on to other organoids, including kidney and lung. And I'm going to stop there, just a little bit ahead of schedule. So the Vivos device is the inspiration and hard work of 2 students, Tiger and Adam. jointly supervised by the three of us, myself, Laurence, and Jeff Rana. A lot of the work in my lab, are people working on various aspects of neurobiology and are using the cerebral organoids in various contexts. There's members of Jeff Lab that have contributed importantly in this project. And here's Amin, who's been working on the microglia. And then just a little small advertisement for the APOC, which is the Applied Organoid Core established in the Donnelly Center to help other labs develop organoid cultures and learn how to do it. Or we can supply organoids. And Javier Hernandez, formerly of Jeff Rana's lab, is the manager. We've collaborated with the NBCC at LTRI to help in the analysis. And I want to thank all the organizations that have funded this project. Thank you.
All right, thank you so much, Lil. We're gonna go to questions now for all three of our presenters from this session. We have quite a few here, but we have some time. So if others have questions they wanna type in, please go ahead. So we'll start with a question for Sam. You there, Sam?
I am, and I'm just starting my video. There you go. Yeah.
Perfect. All right. Thank you. So this question is asking whether you can comment on whether the patterned islet implants vascularize in vivo, and if this is necessary for functional survival and longevity.
Yeah, that's a very good question. So in the in vivo studies in mice where we placed a single fiber into the IP space, there was no vascularization evident after 3 months when we took those fibers out. And the islets were still functional and still viable. And I think that's down to just the size of the fibers. They're only a few hundred microns in diameter. And so I think the cells within those fibers have easy access to the oxygen and nutrients they need to survive. But the caveat there is these are immune deficient animals. And so maybe it's different in immune competent. And if we want to scale the device to large animals and ultimately human patients, there may be a requirement to have some level of vascularization of those devices. So we do have some projects on the go looking at vascularization of larger tissue patches. And Lil's data that you showed on the vascularization of your organoids was fantastic. So you may get an email from me fairly soon to ask.
That'd be great.
Sounds good. Okay, here is one for Liliana. Have you tried growing brain tumors or cancer organoids with the Vivos device?
Yeah, excellent question. Jess Gossio in Jeff Rana's lab is pursuing that. So she implanted some glioblastoma cell lines, or glioma, sorry. Glioblastoma cell lines, and they did form tumors, and she's now looking at implanting patient-derived cancer cells. And I think that would be amazing because it would give us an opportunity to study tumor properties at the very earliest stages, which are otherwise not possible.
Okay, fantastic. Okay, Maria, I had a question for you. I mean, what you talked about is really fascinating, and I'm just curious. You know, with these computational approaches, you know, the sky is the limit because all you need is the computing power. But what are the biggest challenges or the limitations that you do kind of run into as you're developing these types of approaches?
Yeah, I'm not sure the sky is the limit, but we can we can try to push it to move faster, I guess would be than our typical rate. The challenges is for me, because I'm a biologist doing these models, I like to capture as much of the biology as I can. And so it becomes, the difficult part is translating what we know into these algorithms that make sense, not just into any algorithm. The other thing is, the other challenge is when you reach a gap, so like in our cell cycle paper, and when you don't have information, so then there's no experiments to really validate this. So you're just throwing theory out there, which is also interesting. But again, now we're at a point where we don't have information. And so then we're going to try to get experiments to be conducted in order to validate the model. So that's kind of the harder parts.
Yeah, absolutely. And you know, knowing when to simplify and knowing when something's really essential and you're going to lose the information, that certainly makes sense. Okay. We've got a bunch here for Sam. So let's start with this one. Can you comment on the degradability and immunogenicity of the degraded materials?
Yeah, sure. Again, a good question. The materials we use tend to be based on alginate. We're not restricted to only printing alginate, but it's a material that seems to work quite well for us. It doesn't degrade in the body. We cross-link it using cations to form the hydrogel, but there are no enzymes in the body that degrade alginate, and so it's a fairly stable material. And our goal is to have these implants last for, you know, months, if not years, within a patient. And so we don't necessarily want them to degrade. So that's the reason that we're using some of these alginate materials. As far as the immunogenicity of these are concerned, again, we want those materials to act like an immune barrier. So they prevent the physical passage of cells of the adaptive immune system through the shell of these fibers into the cell-containing core. So that you know, that's an important immuno kind of point. But as far as things like foreign body response are concerned, you know, any implanted material that's foreign in the body tends to have that challenge of that foreign body response. And so, you know, that's something that we're working on at the moment to try to reduce that. And with alginate and other materials, there are several strategies that we can take to prevent that.
Yeah, okay, great. And so this question also for Sam relates to liver cell therapy that you mentioned. Are there unique immunological considerations that have to be taken into account for that particular application or indication?
Ooh, that's a good one. Yes, is the short answer. The medium answer is the liver tends to be quite an immune privileged organ. And so if you're putting cells into the liver itself, there's some level of immune privilege. But for ectopic cell transplants that are going somewhere other than the liver, hepatocytes tend to be highly immunogenic. And so, you know, again, that shell of immune protective material becomes very important because it prevents that attack by the adaptive immune system. But the foreign body response is, you know, one of the first parts of that process is attachment of proteins to the implanted device that then triggers the cell attachment by the innate immune system. And the hepatocytes are a source of many different proteins, some of which are involved in the FBR process. So yeah, there's innate and adaptive immune considerations with hepatocytes that are fairly unique to that cell type.
Okay, great. All right, so we have a couple for Lil here. There is one about adapting, I guess, a brain organoid protocol that was developed in Lancaster, and something about you making it more specific and some details around that. Does that ring a bell?
Yeah, more consistent. Yeah. So we published, the protocols published both as a research paper and then a methods paper recently. But basically it all boiled down to very tight control on how you form the embryoid bodies, which is step one. If you control the cell numbers, how they're, the shape of the well that they're put in and the amount of FGF and the timing, if you get, which is within the first few days, if you're careful there, the rest just happens. So the cells know what to do as long as you control the numbers.
Great. And here's another question for you that is an interesting one. So how close are brain organoids to becoming self-aware and developing consciousness?
Yeah, that's not the first time this question has come around. In fact, there have been several scientific discussions that involved biologists, ethicists, neurobiologists, and the conclusion was made that they're nowhere close to self-awareness or consciousness. So we're not there. We don't have to worry about it.
Yeah. Oh, that's fortunate. Yeah.
Yeah.
Interesting. Well, thanks to whoever put that question down. It's an interesting thing to think about. Here's one for Maria. So are you collaborating on particular organ systems or organoid prototypes?
So organs or organoids, no, not yet. Well, Penney is here, and she and I are working on a muscle system, and I think that would be the first organ system that I'm trying to try to understand. With the Nika Shikiba and UBC, I'm trying to create the stem cell culture system. We're trying to create models for that, but not a specific organoid.
Okay. Great. All right. And we've got one more question for Sam here. So for the diabetes approach, how long do you think it's going to take to get to a phase 1 trial? You guys going to do it yourself? Or are you going to partner with a pharma company to do it? Can you say anything about that?
Yeah, sure. I mean, without giving too much away, as far as timing is concerned, I mean, we've got really promising rodent in vivo study data on these implanted pancreatic devices, but we're obviously needing to scale up to large animals to demonstrate that we can scale up to a human relevant size. Those studies are going to be done hopefully towards the end of this year. We'll be having the relevant discussions with regulators accordingly, and then if everything goes well, I think we'd be looking at sometime late 2022 for a clinical trial. That would be our goal. Now, as far as would we partner it out, yeah, I think if the right partner came along and they had the sort of expertise that would be synergistic with our own technology and skills, then absolutely, so a commercialization partner with great reach through to clinicians would be good. But really we're setting ourselves up so that we can take this therapeutic all the way through the clinic to commercialization if it's successful and we have to. So if the right partner comes along at the right time, absolutely we'd consider that, but we're equipping ourselves to go all the way with this.
Yeah, fantastic. I mean, since the last time that I heard the Aspect story, I mean, this kind of internal R&D and doing your own therapeutic development is kind of a new direction, but very exciting. I mean, if you've got a great platform technology, might as well run with it internally. It makes a lot of sense. Okay, so I think that's all the questions that have come through. So thank you to our attendees for typing those out and chiming in. I want to thank one more time all of our speakers. You really made this just a fantastic event. So much cutting edge science, both in the last session and also today's. So thank you for joining us. We wanted to mention this upcoming meeting that will be in Ottawa in November. This is a Tillamooksullik meeting, so very pertinent to what was discussed today. And I believe at Prime, we're also offering some help with registration for this event. So you can certainly contact us about that if you're interested in attending. So with that, thank you, Christine. Thanks to everyone who attended. Thanks to all who helped us organize, and hope to see you very soon. Have a great rest of the day. Bye-bye.
Thanks, everyone. Bye-bye.